Hi guys, welcome to lecture 3 on molecular biology, and today we're going to talk about the genetic revolution. Um, there's been an industrial revolution, and then there's been, I don't know what other kind of things happened in the 60s. Lots of technology revolution. Lately, we had the electronic revolution or the digital revolution, and your generation is going to live through the genetic revolution. And there's some critical and fascinating possibilities that lie ahead for you all. With genetic engineering, we've identified all the genes in the human genome, and that's going to have significant impacts on our ability to fight disease. Um, we can identify the differences that exist between humans. Remember, we're very almost 99% identical, but there are some differences, and of course, these differences give us strengths and weaknesses, and we can take a look at just how they've arisen and how they impact our lives. Um, one of the things that's really fascinating about the genetic revolution is you can actually make changes in people that are alive today and other organisms as well. So not only can we um, maybe fight disease in humans, but we can also fight disease in plants or modify organisms uh, to benefit us, hopefully. Last but not least is the possibility of enhancing human life, and who knows where that's going to lead. People are generally dissatisfied with their lot in life, so who knows? Maybe you could grow an extra finger and you could play your video games better. I don't know the answer is, but this is going to be your chance as a generation to make an impact on human technology. So let's look at some of the common methodologies that are used to manipulate DNA. One of the first, and it's a relatively old technique, is the use of something called phage electrophoresis or polyacrylamide gel electrophoresis. And in biochemistry, you're constantly faced with mixtures of all kinds of different tiny little molecules. And so the role of every biochemist is to isolate and purify specific biomolecules for study. And gel electrophoresis is the way that's done largely. In short, you take your sample and you put it into a gel. It can be made out of stuff like agarose, seaweed. Um, and when you eat Jello, you're eating horse hooves, right? That's what it's made up of, skin. It's the same stuff. It's just collagen. It's a collagen matrix. Anyway, you make up a gel and you make little wells, and then you soak the gel in some fluid, and then you send an electrical charge to that gel. If you look down at the bottom, just below the print here, you can see gel electrophoresis under its process. You load the samples here, and then you put an electrical field through here, and it separates the whatever you put in the wells here. So there's little holes where you put your sample in. And as these samples move through, they get differentiated. That is this. That mixture has all kinds of different size molecules. Some molecules big, some molecules small. The small molecules move quickly, and they separate from the larger molecules. That's it. That's gel electrophoresis. There's also some variations in terms of charge and polarity and the shape of the molecule, but generally its molecular mass is going to be the thing that differentiates its electrophoretic mobility. All right, so I can separate these things. I can separate these components of the molecules, and then I can do analysis. I can actually cut them out of the gel and say, like, hey, what is this stuff? I've purified that sample. Now that's the basis for decoding DNA. And here's a way for us to decode DNA. You can cut segments of DNA, and one of the common tools that's used in phage electrophoresis for DNA analysis is called a restriction enzyme digest. And if I take your DNA and I cut it up with restriction enzymes, these restriction enzymes cut at specific places. Generally speaking, we all have the same kind of patterns that emerge, but there'll be minor variations. And this method allows us to do what's called a DNA fingerprint. So a DNA fingerprint um, for different people will appear differently. And of course, we can use this to identify if you're a murderer. Okay, so if I can get a blood sample from the crime scene and a blood sample from you, I can definitively determine whether or not you've been involved. Of course, that's not all that restriction enzyme digest or DNA fingerprinting is useful for. I can identify certain genes that are present in you and not in other people. Of course, gel electrophoresis is used all the time. If you have a heart attack, they take your blood and they put it in this gel. And if a certain protein's present that's associated with the breakdown of heart tissue, I can say, hey, you had a heart attack, or hey, you didn't have a heart attack. You're just too nervous. So that's phage electrophoresis, and it's used to diagnose all kinds of disease, but it can also be used to digest DNA and make up a DNA fingerprint. Well, that DNA fingerprinting techniques led to a very simple idea of like, hey, let's take a look at the whole human genome, right? So remember that you've got a DNA molecule that's about 3 billion base pairs long. You have 21,500 genes. How do we know that? Well, because they decoded a whole human genome. Now, just imagine how long would it take to count to 3 billion? Your heart beats about 72 beats per second, almost once per second. Um, and you live for about 72 years. And so if you counted your heartbeat for your whole life, you would get to the number, I don't know, 2 billion. All right. So that's how big 3 billion is. It's a very large number. And when I was in college, the Human Genome Project started. I was actually working in genetics laboratories, and we thought it would take about 20 years to do. Well, uh, you know, um, necessity is the mother of invention, and they did the whole process in I think about 5 or 6 years. And there's a you know a famous biochemist named Craig Vetter, and he came up with some um, supercomputers. He bought a bunch of supercomputers, and he bought a bunch of DNA sequencers, and he decoded the human genome. In fact, he decoded his own human genome. And of course, we all have a lot in common with Craig Vetter, but he was the guy that originally said, like, look, right here on chromosome number 1 is a protein associated with glioblastoma, the brain cancer. And here's another gene on chromosome number 22 that's the BRCA breast cancer gene. Um, so you guys are doing your genetic disease and chromosomal mutation projects right now. Um, that information that you're getting about the genetic origin of those diseases has probably has something to do with the Human Genome Project. Anyway, it allows us to, at this point, I think it takes just a few hours to decode a whole human genome. There's been, one super advance after another, and so they can sort of piece together your genetic makeup very quickly. Now that has great possibilities, also has some difficult ethical questions. So, for instance, if you knew that your family had a history of breast cancer, wouldn't you like to know whether or not you carried that gene? And for most of us, the answer is yes, because then you could sort of maybe modify your diet and take care of yourself and get regular checkups, and you might that gene also, if you were to get breast cancer, those genes, when we look at cancers now, we actually look at the genetic makeup of the cancer cells, and it tells us which kind of therapies work best. So, for instance, many chemotherapeutic agents don't really work for that BRCA type or the estrogen sensitive breast cancer gene. So, we don't do that. And so you don't waste time, and you find strategies that work best for you, perfectly detailed for your genetic makeup. So that's a good thing. Um, we can look at the genetic makeup of your baby. So they take cells from the mom's amniotic fluid and can determine whether the baby has genetic mutations. And you could even potentially make changes in the womb, which would be a good thing to do. We'll talk about gene therapy in a second. Um, Anyway, there's a problem with this though. Once that information gets out, who's going to use it? So let's say you wanted to get health insurance and the health insurer found out that you were likely to get breast cancer. They might want to charge you more money. Or maybe there's a gene associated with alcoholism or mental health issues. Would your employer like to know about that? And that's one of the sort of a path of ethical dilemma for this genetic technology. Well, what do you do if you do know you have a problem? And that walks us down into a branch of medicine called gene therapy. Now, it's been in the works for about 20 years now. There's not any currently available, easily available gene therapies, but they're coming. And one of the recent advances that will allow for gene therapy miraculously is a process called CRISPR. And CRISPR stands for Clustered Regularly Interspersed Short Palindromic Repeats. And CRISPR-Cas9 is a protein found in bacteria. And it identified viral bacteria. So remember, bacteria can become infected with viruses, those bacteriophages. And so viruses or bacteria have like some cool proteins that help destroy viral bacteria or viral DNA. Now, we've taken that bacterial machinery and modified it to do things to human DNA. And so the procedure is called CRISPR-Cas9, and it allows you to make very specific cuts in very specific places. And you can cut things out and you can add things in. And that's the silver bullet for genetic engineering, all right? So it's easy to get a code. You can literally make a phone call or order, just like you order in Chinese food today. You can say like, hey, I want a sequence of DNA that goes AAATTTGGGGC, whatever your code you want. And in a day or two, you'll get a little bottle full of DNA that has that code in it. And you could insert that into cells. And that is what gene therapy is all about. So if you have a genetic disease, say cystic fibrosis or muscular dystrophy, you could potentially introduce the correct sequence of DNA into those cells that are malfunctioning. Of course, the problem with a disease like cystic fibrosis or muscular dystrophy is that, you're made up of like 70 trillion cells. So how do I get DNA into 70 trillion cells? Well, one of the answers is the time to do it is when you're an infant. So when you're an infant, you're made of one cell, or maybe you're made of whatever, 300 cells. If I could do CRISPR gene therapy, then I could fix you. But we'll see. The technology rapidly advances. And I guess what I'd like you guys to think about is what's next. It's part of your project is to entertain the possibility of ways to treat the diseases that you're studying. So think about that. In fact, there's probably gene therapies on the horizon. It's one of the questions I ask you to answer. So that leads us to the big question. What is next? Well, look, this definitely is going to enable the cures for genetic diseases, right? Because we can modify genes. But guess what? All disease has a genetic basis. We're all more or less prone to get cancer or heart disease or diabetes, and these are the big killers of Americans in 2020. But those things can be erased. Obesity is one of the biggest epidemics our country is. I think something like 40% of Americans are obese at this point. You could potentially take a drug and cure obesity. Of course, that's going to lead to life extension. Not only just living longer, but maybe even having a higher quality life. As you age, all kinds of things happen. Your skin gets saggy, your hair falls out, you lose your muscle mass, you become miserable. Well, you could potentially have genetic engineering that would mitigate that. Of course, the biggest thing for you guys is to ask yourself, well, what am I going to do with my life? And the genetic revolution is going to include you whether you like it or not. How much would you pay for the ability to be healthy for the rest of your life? Or, hey, how much would you like to earn working in that industry? So that's something to think about. And you can do it. You can become the next great genetic engineer. So there you go. That is the end of molecular genetics. See you.